Good day, everyone. A group composed of Estrella, Nuevas, and yours truly is tasked to critique and report about our chosen paper titled Arrayed CRISPR Reveals Genetic Regulators of Tau Aggregation, Autophagy, and Mitochondria in Alzheimer's Disease Model. The objectives of this presentation are, first, to discuss the findings and implications within our chosen journal article. Second, to identify the strengths and weaknesses of the research performed within the study. And lastly, to provide recommendations that can help improve and enhance the selected article. Alzheimer's disease, or AD, is the most common type of neurodegenerative disease characterized by misfolded and abnormally structured tau proteins. The normal tau proteins form part of a structure called the microtubule. One of the functions of the microtubule is to help transport nutrients and other important substances from one part of the nerve cell to another. Hence, anomalies in the said protein can cause a wide array of diseases including AD. Tau proteins serve as the main target of therapeutic drugs for the disease. Yet, while they have promising results, they still have limited efficacy and are restricted by toxicity, which can be attributed to the blood-brain barrier, which limits access to the tau tangles. Nonetheless, a handful of methods including RNA interference, open reading frame or ORF, and pooled CRISP methods are useful in viewing the genome sequence for potential AD targets. Yet, they still offer limited results due to challenges such as difficulty in capturing morphological features, short target inhibition, etc. However, through an arrayed CRISPR format which overcomes the aforementioned limitations as well as offer high imaging content, it was discovered that a network of genes involved in immune response modulated tau aggregation including the NFKB pathway and the LKB1 complex, hence offering novel targets for AD drugs.
Now, let us cover the methods used in this study. The methodology of this study can be simplified into three key processes. First, we have cell culture model of Alzheimer's disease. Second would be genome-wide CRISPR screening. And third is the validation of findings. For the first step, the researchers used a human cell culture model of Alzheimer's disease, which involves inducing tau aggregation and mitochondrial dysfunction in the cells. For the second step, the researchers employed a genome-wide CRISPR screening approach to identify genes that regulate tau aggregation, autophagy, and mitochondrial function. They used a technique called Arrayed CRISPR to target each gene in the genome one by one and assess its effect on the three processes of interest. Under these three processes are a CRISPR-Cas9 editing system, high-throughput screening, and Arrayed CRISPR library. The CRISPR-Cas9 system is an editing tool that is used to introduce target mutations in genes of interest. The researchers designed guide RNAs or gRNAs to target specific genes and then used lentiviral vectors to deliver the gRNAs and Cas9 protein to the site. For high-throughput screening, the study used high-throughput screening approach to test the effects of your CRISPR-Cas9 induced mutations on various cellular processes. The researchers used an automated microscope to analyze the cells and identify changes in tau aggregation, autophagy, and mitochondrial function. The study also used an arrayed CRISPR library, which contained individual gRNAs targeting over 500 genes associated with Alzheimer's disease. The library allowed the researchers to test the effects of individual gene knockouts on the cellular process of interest. Lastly, for the validation of findings, the researchers validated the results using a variety of experimental approaches including Western blotting, immunofluorescence microscopy, and functional assays. Immunofluorescence staining is a method that they used to visualize tau aggregation and autophagy. The researchers utilized antibodies specific to tau and autophagy markers to label the cells and then image them using a fluorescence microscope. To further elaborate on the different methods conducted by the researchers, the following are some of the processes performed. There's cell culture and lentiviral arrayed CRISPR screen. There's high-content imaging, immunochemistry, tau aggregation induction and detection, autophagy detection and compound treatments, SHSY5Y neuronal differentiation and lysosome tracking, organelle labeling and membrane potential assessment, Sanger sequencing and tide analysis, and as well as SIRNA and ORF processes. Moreover, some of the elaborated methods include mitochondrial seahorse assays, total RNA isolation, RNA-QC and cDNA preparation, quantitative PCR or qPCR and data analysis, RNA-seq analysis, and last but not the least is bioinformatics analysis. In performing the aforementioned methodologies, the researchers were able to identify a new set of genes to regulate tau aggregation, autophagy, and mitochondrial function, and second, determine new connections between the processes involved in Alzheimer's disease formation. The first result of this study involves gene identification. From the study, the researchers identified several new genes that promote or inhibit tau aggregation in Alzheimer's disease. These include TUBB4A, RAB31, and CLUH. Among these three, TUBB4A in particular was found to be a novel regulator of tau aggregation and a potential therapeutic target. In terms of regulation of autophagy, the study also identified several new genes that regulate such process. These include ZFYVE27, FAM134B, and RB1CC1. Among these, ZFYVE27 was found to be a negative regulator of autophagy and a potential target for promoting autophagy in Alzheimer's disease. In terms of regulation of mitochondrial function, the researchers identified several new genes involved in this process. These involve ELOVL5, CRS2, and MF2N. ELOVL5 was found to be a positive regulator of mitochondrial function and a potential therapeutic target for Alzheimer's disease. For the second result of the study, the researchers were able to discover connections between the processes involved in Alzheimer's disease formation. Here, they were able to discover that the regulation of tau aggregation is actually affected by mitochondrial proteins, and there is also a role of autophagy in regulating mitochondrial function. It can be seen here in this graph that there are interconnectedness between these different genes, as well as this map that includes all the different genes involved in Alzheimer's disease, which are kind of all connected when you see in this specific figure.
Let us now discuss the three major points found in the paper and their implications.
Firstly, the study examined the link between immune cell activation and inflammation pathways to tau aggregation. The authors found that inhibiting NFKB, which is a transcription factor most commonly associated with immune cell activation, led to a decrease in tau aggregation, highlighting a potential target for intervention in Alzheimer's disease. Additionally, the study demonstrated that genes linked to inflammation can modulate tau aggregation in response to a pro-inflammatory extracellular environment. These findings are significant as they suggest a potential therapeutic approach to treating Alzheimer's disease by targeting the inflammatory response.
Secondly, the study highlighted the role of autophagy in the regulation of tau aggregation in Alzheimer's disease models. The authors identified several genes and pathways associated with autophagy that influence tau aggregation, with knockdown of certain genes resulting in increased autophagy and decreased tau aggregation. Furthermore, the authors observed that disrupting PI3K genes led to increased tau aggregation, indicating the importance of basal autophagy in regulating tau aggregate clearance. The study also demonstrated the role of the LKB1 complex in modulating tau aggregation, indicating the concerted action of tau kinases and autophagy in regulating aggregation propensity and clearance. These findings suggest that targeting autophagy could be a potential therapeutic strategy for treating Alzheimer's disease.
Finally, the study investigated the influence of genes on mitochondrial function in AD models. The authors identified several genes that affect mitochondrial morphology and function, with knockdown of certain genes resulting in decreased mitochondrial function and increased tau aggregation. These findings suggest potential therapeutic targets for the treatment of AD. This study also revealed a link between mitochondrial morphology and distinct cellular functions, such as glycolysis, indicating the importance of mitochondrial function in Alzheimer's disease. Overall, the study by Duan et al. Suggests several potential therapeutic targets for the treatment of AD through targeting inflammation, autophagy, and mitochondrial function. These findings have important implications for understanding the pathogenesis of Alzheimer's disease and developing novel therapies for this neurodegenerative disease. The study employed the highly advanced CRISPR-Cas9 gene editing technology to systematically identify genes linked to tau aggregation, autophagy, and mitochondrial function in AD models. This approach enabled the precise identification of key genes that could serve as potential therapeutic targets for AD. Moreover, the use of the arrayed CRISPR method allowed for a more comprehensive and systematic approach to identifying genetic regulators of complex cellular processes, including tau aggregation. The study's findings offer valuable insights into the underlying molecular mechanisms of AD. In particular, the identification of key genetic regulators involved in the associated pathways provides an understanding of the intricate cellular processes contributing to AD's development and progression.
One weakness of the study is its heavy reliance on the CRISPR-Cas9 method. While this gene editing process has shown promising results, including multiplexing, cost-effectiveness, simplicity, and accuracy, it is still a limited technique in terms of efficiency. In specific, it lacks on-target editing efficiency, incomplete editing, and inaccurate off-target editing. That said, this could result in unwanted mutations in the embryonal level, affecting the human cell lines and the future generations. Likewise, the delivery factors of the CRISPR method imposes potential immunogenic toxicity. It can cause large rearrangements of DNA through retrotransposition to theoretically trigger tumor development, according to a paper published in New York Communications. It can also increase the risk of cancer. Therefore, despite its capacity to be of use in the development of drugs for Alzheimer's disease, it causes a cascade of new problems which compromises the safety of the individuals and its efficacy in avoiding unwanted genetic alterations. It is questionable ethically and in the long term. Moreover, another weakness of the methodology is the induction of lentiviral vectors. This is questionable due to its potential immunogenicity and cytotoxicity. The lentiviral vector can disrupt the normal regulation of the cell development and proliferation, leading to oncogenesis in the infected cells due to the disruption of the expression of tumor suppressor gene. Also, mismanagement can cause inflammations since lentivirus are pathogenic to humans. Given the weaknesses discussed earlier, it is suggested that a comparative analysis of the efficiency of CRISPR and lentiviral vectors with other gene editing methods should be done to ensure that the methodology is reliable. There are currently existing alternative techniques in gene editing, one of which is TALEN, which is recently discovered by a research team from the University of Illinois. It is believed to be up to 5 times more efficient than CRISPR-Cas9 in a highly compact form of DNA called heterochromatin. It also provides more freedom and flexibility in target site selection compared to CRISPR. Moreover, unlike CRISPR, which can introduce multiple gene mutations concurrently with a single injection, TALENs are limited to simple mutations which would result in less harm and instability to the genome. Another alternative is prime editing. This technique is specifically designed to overcome some of the limitations of CRISPR. It was found out that unlike CRISPR, prime editing doesn't rely on the ability of a cell to divide to help make the desired changes in the DNA. That means it could be used to correct genetic mutations in cells that often don't divide, such as cells in the nervous system.
In conclusion, the study entitled Arrayed CRISPR Reveals Genetic Regulators of Tau Aggregation, Autophagy, and Mitochondria in Alzheimer's Disease Model was able to reveal that there are several genes involved in such processes, and that they are interconnected with complex interplay between them. In this, the study highlights how the identification of these novel regulators could actually lead to the development of targeted therapies that can either slow down and hopefully prevent the progression of Alzheimer's disease. To summarize, the final takeaways from this study are: first, the use of arrayed CRISPR screening is actually an effective approach to systematically identify genes that regulate key processes in Alzheimer's disease. Second, the identification of novel regulators of tau aggregation, autophagy, and mitochondrial function could lead to the development of new therapies for this disease. And last but not the least, the findings of this study open up new avenues for further research into the genetic mechanisms of AD and the development of targeted therapies that can be developed for future treatments. Going back to the objectives we mentioned during the introduction, we were able to successfully achieve all of them. As first, we discussed the study's findings and methodology. Second, we identified the strengths and weaknesses of the paper. And last but not the least, we provided possible recommendations and gaps that can be focused on for future studies. Now, what is the significance of this study to our course in human biology? Well, the significance of gaining knowledge regarding the activity and identification of the regulators for Alzheimer's disease is beneficial for human biology students for the reason that, as future medical practitioners, we have the duty to provide the best set of care and service that our patient and community needs. Through exploring potential means of therapy and widening our knowledge with regards to the mechanism of diseases, we will be able to further think, innovate, and create new and breakthrough ideas that can hopefully stop and cure Alzheimer's disease in the future. By recognizing how disease pathology works and incorporating it into the subjects of anatomy and dealing with these kinds of diseases, we can aid in brainstorming ideas and potential solutions that can enhance and uplift the treatment for patients with Alzheimer's. To wrap up our presentation, once again, I am Arrayed Estrella, and together with me are my groupmates Adrian, Gabriel, MIRASOL, and Jana Veron Huevas. Thank you for listening.